Good evening and welcome you all to PSI Online Webinar 15. Being a life member of the Physiological Society of India and a co-opted member of the Education Committee of the Physiological Society of India, I am really privileged to participate in the PSI Online Lecture Series 15 as a moderator of this webinar. The Education Committee of the PSI is trying to cover the system-wise different topic in each webinar. These lectures are scheduled on the last Saturday of each month. In the last few lectures, we have already covered different parts of general physiology, immunology, endocrinology, excretory system, and nerve muscle physiology also. In continuous with our previous lecture on the action potential by Dr. Bhatt, as a part of nerve physiology, now we are going to introduce our today's speaker, Dr. Bithin Kumar Maji, who will illuminate on the EC coupling from the cellular mechanism to therapeutic targets. Dr. Maji is the Associate Professor, Department of Physiology, Serampur College, affiliated to University of Calcutta. Dr. Maji earned his master's degree in physiology with a special paper in biochemistry from University of Calcutta and the doctoral degree in science from physiology, particularly from the subject physiology from Jadavpur University. After that, he joined the Department of Physiology, Serampur College as a lecturer in 2003, and currently he is working as an associate professor, Department of Physiology, Serampur College, postgraduate course coordinator of human physiology under University of Calcutta, Serampur College campus. In addition to that, he holds the post of vice principal, IQAC coordinator, and the chairperson of the Institutional Animal Ethics Committee at Serampur College. Dr. Maji has 28 plus years of experience in both teaching and research. He has acquired government funding from UGC and ICMR to pursue his research. He has published his research article in different well-reputed international and national peer-reviewed journal with high impact factor. Now we are eagerly waiting for your lecture, sir. Dr. Maji, the platform is yours now. Sir, please.
Thanks, Dr. Banerjee.
Welcome. Yes.
Good evening to everybody. I am thankful to the PSI Education Committee and office bearers, all the office bearers of the Physiological Society, and I am thankful to the PSI for choosing me as one of the speaker of PSI Oration Lecture. The today's topic is EC coupling from cellular mechanisms to therapeutic targets. In the last lectures, the speaker actually highlighted the generation of the action potentials, and this lecture is linked with the action potentials of the nerve fibers. Now, action potential, as well as when transmits to the muscle fibers, it causes the excitation contraction coupling. And this EC coupling is a fundamental physiological process that links the electrical stimulation of muscle fibers to their contractions. It begins with the generation of an action potential in a motor neuron, which triggers the release of acetylcholine at the neuromuscular junction. This neurotransmitter depolarizes the muscle membrane, leading to the opening of voltage-gated calcium channels in the transverse tubules. The resulting influx of calcium ions from the sarcoplasmic reticulum initiates muscle contraction through interactions between actin and myosin filaments. Understanding excitation contraction coupling is crucial not only for basic muscle physiology but also for identifying therapeutic targets in various neuromuscular disorders, heart disease, and muscle pathologies. Potential therapeutic interventions may include calcium channel modulators, acetylcholine receptor enhancers, and gene therapies aimed at restoring normal excitation contraction functions. By exploring the intricate mechanism of excitation contraction coupling, researchers can develop innovative treatments that address the underlying causes of muscle dysfunction and improve patient outcome. And these are my outline of the lectures, and I am going to one after one. Before going to the excitation contraction coupling, each and everyone have to know the basic muscle physiology and the structural organization of the muscles, and followed by the process of excitation contraction coupling. Now, the muscle cells, like the neuron, is an excitable tissue. That means an action potential is generated chemically, electrically, and mechanically. And muscle is a contractile tissue where the chemically stored energy transformed into a mechanical energy. If we carefully study the uh, microscopic structure of the muscles, there are three different types of muscles. One is the skeletal, another is the cardiac muscle, and another is the smooth muscles. As the skeletal muscles, we found that there is a regular striations, cross striations. That is why these are called the striated muscles. Cardiac muscles also we find the striations, and in case of the smooth muscles, and there is no such striations, and that is why the skeletal muscle and the cardiac muscles are considered as the striated muscles, and the smooth muscles are non-striated muscles. On the other hand, if we study the action of the muscles, some muscles are called the voluntary muscles. That means these muscles actually we can modulate or contract according to the willpower of an individuals, where the skeletal muscles is considered as the voluntary muscles, and but the cardiac muscles and smooth muscles are the involuntary muscles. And there are special characteristics between the voluntary muscles and involuntary muscles are there. In case of the control, in case of voluntary muscles under conscious control, but in case of the involuntary muscles operate without conscious effort. And if we consider the location, the voluntary muscles are attached to the bone, that is, these are the skeletal muscles. On the other hand, involuntary muscles found in the internal organs, that is the smooth muscles. As the term we mentioned the smooth muscles as because there is no such cross striations, that is also called the plain muscles. And if we consider the function, the voluntary muscles enable intentional movements. On the other hand, involuntary muscles regulate autonomic bodily functions. Voluntary muscles actually controlled by the somatic nervous systems. On the other hand, the involuntary muscles controlled by autonomic nervous system. And so these are the important characteristics between the voluntary and the involuntary muscles. Now, the skeletal muscles, as the name indicates, are attached to the bones of the body skeleton, and their contraction results in the body's movement. And they constitute about the 40% of the total body mass of an individual. Now, if we consider the functional anatomy and the organizations, each muscle fiber is surrounded by delicate connective tissue called the endomysium, which contains a large quantity of elastic tissue arranged longitudinally. The muscle fibers are grouped into several bundles called the fasciculi. Each fasciculus is surrounded by a stronger sheath of the connective tissue, which is known as the perimysium. And all the fasciculi collectively form the muscle belly. The connective tissue that surrounds the entire muscle belly is called the epimysium. And tendons are the fibrous terminals ends of the muscles made up of a collagen fiber. So if you see that this is the structure of a muscle belly where the this portion is called the muscle fiber and the outermost layer is called the endomysium. When muscle fibers actually grouped to form a fasciculi, these are the fasciculi, and this portion are the connective tissue, and the outermost layer is called the perimysium. And when all the fasciculi actually clubbed together or grouped to form a muscle belly, and the outermost layer is called the epimysium. And if you see that, so I have already mentioned that it is a muscle belly. Actually, it consists of various types of the number of the fibers, and each of the fibers actually consists of the different types of the fibrils, and these are the arrangements of the thick and the thin filaments, and the filaments actually contains various types of the protein. These are called the muscle proteins. And if we study the structures one after one, the muscle fibers are the long cylindrical and the multinucleated cells, and the membrane of the fiber is called the sarcolemma, and the cytoplasm of that cells are called the sarcoplasm, and the myofibrils actually the embedded in the sarcoplasm. The sarcolemma and the sarcoplasmic reticulums from the sarcotubular system, which is an important system actually linked with the excitation contraction coupling. If you see the structure that the muscle fiber contain numerous myofibrils and arranged parallelly, each myofibrils comprises thick filaments made of contractile proteins already mentioned. There are two types of the contractile proteins. One is the myosin and the actin. These are called the contractile protein as because they are directly involved with the muscular contraction, and this is the distribution pattern, that is the 1500 per myofibril, and the thin filaments where the 3000 protein actually or actin molecules per myofibril. So these are the structural organization where this is the muscle fiber, and this is the fibril and the filaments arranged in such a way. Now, the peculiar arrangement of these myofilaments seen under the light microscopes gives an appearance of the alternate dark and light band, which is called the striations. These striations are nothing but the different types of the refractive indexes, and that is why it looks like some portion is the dark and some portion of the lighter portion. If you carefully see the structure, that the arrangement of the various types of the proteins, this total length is called, there are the various types of the band, that is called the A band, and A band is actually nothing but the consists of the myosin proteins, and some portion of the A band actually overlap with the I band. This is the I band portion, this is the lighter portion, and the midline of the I band is called the Z line, and the distance between the two Z lines are called the sarcomere, and the sarcomere is the structural, functional, and the fundamental unit of the muscular contractions. Now, if we consider the various types of the proteins actually involved in the excitation contraction couplings, there are three different types of the proteins. I have already mentioned that the myosin and the actin are the contractile proteins. On the other hand, tropomyosin and the troponin are considered as the regulatory proteins. Sometimes we mention the term the relaxation proteins. And another proteins which we call the anchoring proteins, say for example the actinin, titin, nebulin, and the dystrophin. So these are the various types of the proteins actually clubbed together to form a muscle structure. So this is the structural organization where the thick filaments forming the myofibrils and rebuilt by the electron micrographic structure. Now, if we carefully study the structure of the myosin molecule, myosins are superfamily of the ATP-dependent actin-based motor proteins, and the myosin molecules and a heavy chain having a tail portion. This is the tail portion hinge. That is, this portion is the hinge and the head domains, and a light chain presents near the myosin head that modulate calcium-dependent transduction of force by the myosin molecule. And this is the structure of the myosin molecule, and arrangement of the myosin molecules in the thick filament in this way. And I have already mentioned this is the tail portion. This is the tail portion. Okay, sorry. This is the tail portion, and this is the head portion. And sometimes the myosin contains the one head we call the myosin 1, and sometimes we found the head, two heads, that is called the myosin 2 molecules with two heads. Now, if we study the digestion of the myosin molecules with the trypsin and the papain, we find that when we treat the myosin molecules with the trypsin, such fragments are occurred, which is called, one is the light meromyosin, and upper portion is called the heavy meromyosin. When the heavy meromyosin is treated with the papain, actually we again obtain the two fragments. One upper part is the head part, which is called the heavy meromyosin S1, and the lower part, that is the only tail portion, that is the fibrous heavy meromyosin S2. So on the other hand, the thick filament have myosin molecules oriented in opposite directions. I have already shown in this figure. This is the opposite direction. Half of the myosin head face one way and the other half face the opposite. This arrangement results in a central region without myosin head, forming the lighter edge zone in the A band. I have already shown in the last slide. Overlapping myosin molecules create a central bulge in thick filaments. This bulge corresponds to the M line observed under light microscopy. And in case of the thin filaments has a head end and a tail end that is anchored to the Z line. Each thin filament is made of the contractile protein molecule, that is the actin, and two types of the regulatory protein, that is the troponin and the tropomyosin. This globular structure is called the actin, and this portion is this globular structure where mentioned ICT is the troponin, and this is the tropomyosin, that is the filamentous protein. This is the structural organization where the actin, myosin are and the tropomyosin arranged in this way. About 300 to 400 actin molecules are present in each filament. The actin molecules form a long double helix consisting of the two chains of globular units in this way, and the globular molecules are called the G-actin. That means the individual molecules are called the G-actin. When all the G-actins actually form a chain-like structure, this chain is called the F-actin molecules, and this structure shows the arrangement of the actin molecule, and these are the head of the myosin molecule. Now, tropomyosin, I have already mentioned that tropomyosin is a filamentous protein. About 40 to 60 tropomyosin molecules are present in each thin filament. Tropomyosin are long filaments that lie in the group between the two chains of actin molecules. It covers the binding site of actin where the myosin head comes in contact with actin. Thus, it is a regulatory protein that prevents the interaction between the actin and the myosin filaments. On the other hand, troponin molecules, there are troponin molecules is a globular protein. It has three components on either troponin T, troponin I, and the troponin C. Troponin T binds with the troponin tropomyosin, and the troponin I prevents the interaction of the myosin heads with the active sites of the actin, and the troponin C contains the binding sites of the calcium ion that initiates the muscular contractions. And each molecule of troponin C binds to 4 molecules of calcium ions. And these are the anchoring proteins. Anchoring proteins plays an important role and these proteins actually interact with each other to form a club-like structure where actin, myosin, troponin, and the tropomyosins arranged in a compact structure. Alpha-actinin cross-links the actin filaments in the area of Z-line, whereas the titin, earlier called the connectin, or the gap filaments, is a large elastic filament which interconnects the jet line. It forms the series of elastic component of the muscles. On the other hand, nebulin is the another connect actually the anchoring protein is connected at one end to the alpha-actinin in the area of the jet line and at another end to the troponin tropomyosin complex of the thin filaments at regular interval. So these are the structure, and this is the titin molecule, and this is the alpha-actinin, this structure, and this protein actually called the dystrophin. So all are the anchoring proteins arranged in this way. So the dystrophin glycoprotein complex forms the best known anchor protein complex, which provides structural support and strength to the myofibril, actually shown in this figure. Now, sarcotubular system is an important system. I have already mentioned that sarcolemma, along with the sarcoplasmic reticulum, actually of the muscle, forms the highly specialized system called the sarcotubular system. And this sarcotubular system, actually, these are the various types of the tubules. This is called the T tubules, and this is the longitudinal tubules having two cisterns. This is the longitudinal tubules of the sarcoplasm, and this is the terminal cistern. So one transverse tubules and the two cisterns joins together to form a triad, and it opens at the ZAI junction. So this is the diagrammatic features of the sarcotubular system. Now, the muscle is an excitable tissue. I have already mentioned that means the action potential is produced when stimulated. The skeletal muscle actually responds to stimulus by contracting. That means it is the mechanical phenomenon or events. The events that links the electrical phenomenon with the mechanical phenomenon are called the excitation contraction phenomenon. These three phenomena which marks the excitability and contractility of the muscle when stimulated by the nerve innervating it. Now, now I am coming to the actual events of the muscular excitation. It refers to the electrical phenomena and ionic fluxes in the skeletal muscle fibers. The transmission of the across neuromuscular junction and end plate potential is developed at the motor end plate by the nerve impulse traveling across the innervating axons. Then end plate potential is localized and non-propagated. However, when end plate potential reaches a threshold level, it produces an action potential that propagates over the muscle fiber surface and into the muscle fibers along the transverse T tubules. And the essential features of the electrical phenomenon that occurs in the muscle fibers, both resting membrane potential and the action potential, are similar to those occurring in a nerve fibers. So these are the quantitative differences between the electrical phenomena occurring in the skeletal muscle and the nerve fibers. If you study that, the resting membrane potential of the muscle minus 90 millivolt, but in case of the nerve, it is minus 70 millivolt. On the other hand, initial excitation threshold level in case of muscle 30 millivolt, but in case of the nerve, it is 15 millivolt. The magnitude of action potential in case of the muscle 120 to 130 millivolt, but in case of the nerve, it is 100 to 105 millivolt. Duration of the spike 5 milliseconds, in case of the nerve 0.4 to 2 milliseconds. The absolute refractory period of muscle is 1 to 3 milliseconds, but in case of muscle, it is 0.4 to 2 milliseconds. And the maximum number of impulse 100 to 200 per second in case of the muscle. In case of the nerve, it is 1000 per second. And the chronaxi, actually, which is the one of the important parameter of the excitability, which is longer in case of the muscle but shorter in case of the nerve, and the conduction velocity of the action potential 3 to 5 meter per second in case of the muscle, but in case of the nerve it is 120 meter per second. And these are the equilibrium potential different ions in the skeletal muscle and the nerve fiber, that is the sodium, potassium, hydrogen, chloride, and the bicarbonate ions. Now, what is excitation contraction coupling? Actually, excitation contraction coupling refers to the series of events that link to the action potential of the muscle cells membrane to muscular contractions. Although EC coupling in the myocardium is similar in many ways to the skeletal muscles and the smooth muscles, there are also critical differences. Now, in resting condition, it is the mechanism of release of the calcium ion from the terminal systems of the longitudinal tubules. In resting condition, the calcium release channel remains closed due to the mechanical interlocking between the dihydropyridine and the ryanodine receptors. So this is the dihydropyridine receptor, dihydropyridine protein, and this is the ryanodine. It is interlocked. During the activation state, that is the depolarization of the T tubules, a conformational change occurs where the dihydropyridine results in the opening of the ryanodine receptor and release of the calcium ions. So if you carefully observe that, this is the resting state, and when the action potential actually comes into the T tubules, what happens? This lock is open and the calcium comes out. Now, if we consider that the molecular basis of muscular contraction, the A.F. Huxley and the H.E. Huxley in the year of 1954 put forward the sliding theory or ratchet theory to explain how the actin filament slides over myosin filaments, forming the actin-myosin complex during muscular contraction. So this theory is called the walk-along theory or the modern theory of the muscular contraction. This theory explains that sliding of filaments is brought about by a repeated cycle of formation of the cross bridges between the head of myosin and the actin molecules. So this is the cross bridge cycles. At resting condition, the myosin binding site on the actin is covered by the troponin tropomyosin complex. I have already mentioned that the troponin tropomyosin complex is also called the relaxing protein or regulatory protein, as because at normal condition, that means when there is no excitation, what happens? The myosin binding site is covered by the tropomyosin molecules. But on excitation, the calcium actually ion, which is actually released from the sarcoplasmic reticulum, binds with the troponin C molecule. This is the troponin C molecule. And this results in the conformational changes and lateral displacement of the tropomyosin takes place. So this is this figure shows the resting condition, and after binding with the calcium, a three-dimensional changes occurs in such a way that lateral movement of the tropomyosin takes place, resulting now the myosin binding sites is now open. And as the myosin binding sites is now open, the head of the myosin, actually myosins 2, there are two heads. One head is responsible for binding with the myosin, and another head actually contains an enzyme which is the myosin ATPase enzyme. So as the lateral movement of the tropomyosin molecule takes place, the myosin head actually binds with the actin molecule. So this is if you carefully see the structure, so this is the two jet line, this is the sarcomere structure, and this line indicates the actin molecule, and this red-like structure indicates the myosin head. And initially, this is the formation of the actin-myosin complex. That means the attachment of the myosin head to the active site of the actin filament. So this is the attachment takes place. This is the stage 2. Now, when the attachment takes place, what happens? The ATP molecules actually splits to form the ATP and the PI, and the energy is generated. And the energized myosin head actually pulled uh, inside, and this phenomena is called the power stroke, resulting. And this is the myosin head binds with the molecules, and in the stage 3, detachment of the head of the cross bridge from the active side of the actin filament takes place. And lastly, the reactivation of the myosin head takes place. So these are the 5 stages of the cross bridge cycling. Now what happens? Now in this figure, I want to show that the steps of the muscle relaxations. So as soon as the contraction takes place, now what happens? Now calcium ion actually, which actually released from the sarcoplasmic reticulum during the contraction, again back to the sarcoplasmic reticulum by a calcium pump. And that means the transport of the calcium ion from the sarcoplasmic reticulum into the longitudinal tubules by the calcium pump takes place. On the other hand, hydrolysis of the ATP by myosin energize the cross bridges, providing the energy for force generation. Binding of ATP to myosin causes dislocation of the cross bridges, allowing the bridges to repeat their cycle of activity. Hydrolysis of the ATP by calcium ATPase in the sarcoplasmic reticulum provides the energy for the active transport of the calcium back into the terminal cistern, lowering the cytoplasmic calcium, ending the contraction, and allowing muscle fiber to relax. Now, if you carefully study the sarcomere structure, the changes produced by sliding thin filaments over the thick filaments during the muscular contractions. So these are the Z lines, 2 Z lines, and after contraction, what happens? 2 Z lines move to each other. That means the shortening of the length takes place. So changes produced in a sarcomere by sliding of the thin filaments over thick filament during the muscular contraction. Here, A, the upper portion of the figure is the relaxed state, and lower portion, that is the B, this is the contracted states, and Z line actually moves to each other, and thereby shortening of the sarcomere length takes place. Now, if we carefully study the sequence of the events during the muscle contraction and the relaxation when stimulated by a nerve, that means from the nerve excitation, the stimulation of the motor neuron, followed by initiation of the action potential in motor neurons action, that means the nerve conduction, and this propagation of the action potential in the motor nerve, followed by impulse reaches at the nerve endings, that means at the synaptic button. What happens? The neuromuscular transmission, increased permeability of the presynaptic membrane to the calcium ions, inflow of the calcium ions from the extracellular fluid into the nerve terminals, followed by release of the acetylcholine from the microvesicles present at nerve terminals, followed by diffusion of the acetylcholine into the synaptic cleft, binding of the acetylcholine receptors on the motor end plate, and opening of the calcium gated channels in the motor end plate membrane, then entry of the mainly the sodium ions, and to a lesser extent of the calcium ions through these channels into the muscle fibers, and the development of the end plate potential and the muscle excitation. Local end plate potential, when reaches a threshold magnitude, a voltage gated sodium channels are opened up at the site, followed by generation of the action potential in the muscle fibers by the end plate depolarization, and the propagation of the action potential in the muscle fibers along the surface into the fiber along the T tubules, and the excitation contraction coupling takes place. That means the release of the calcium ion from terminal cistern, then diffuse diffusion of the calcium ions into the sarcoplasm, and the binding of the calcium ions to the troponin C, I have already mentioned, and the muscular contraction takes place according to the molecular theory. The uncovering of the binding sites of the myosin on actin, followed by cross bridge formation between the myosin head and actin, and the angular movement of cross bridge, that is called the power stroke, and the sliding of the thin filaments over the thick filament takes place, and which is initiate the muscular contraction. And during the muscular relaxation, what happens? The active transport of calcium ion into sarcoplasmic reticulum, followed by decreased concentration of the calcium ion in sarcoplasm, followed by removal of calcium ions from the troponin C, and the cessation of cross bridges cycles, as because the after detachment of the calcium ion from the troponin C, and the tropomyosin molecules again back to its position, and the muscle relaxation of the muscle fiber takes place. Now, I like to mention that there are some alteration in the excitation contraction coupling can have numerous origins because of the complex structure of the triad and many proteins involved in the excitation contraction coupling and calcium homeostasis within muscles. The different types of alterations can be clustered schematically into four groups. One is the direct mutation in a protein of the CRC. Another is the alteration in the triad structure. Third one is the alteration in the regulation of calcium channels. And fourth one is the alteration in calcium stores. Now, this is the possible origins of alteration in the EC coupling. Now, what happens in the this is the event one. The mutation can occur in one of the calcium release complexes, that is the CRC proteins, mainly ranitidine receptor 1 and the dihydropyridine receptor. The arrows represents the calcium fluxes, which mutation in the channels can therefore modify. Now, number two, mutation can occur in proteins involved in triad formation or maintenance, resulting in modification of triad shape or in the triad lipid composition or the triad targeting CRC proteins. Next, the alteration in the redox state, ROS or RNase of the muscle fibers can alter ranitidine receptor 1 regulation by direct mutation in a protein involved in redox regulation or by indirect increase in the oxidative stress. Ranitidine receptor 1 regulation can also be altered by modifications in splicing, for example, as a result of sequestration of muscle blind proteins or the splicing regulators. And the stage 4, the calcium stores can alter a result of mutations in the proteins involved in their refilling. Now, these are the therapeutic targets. Number one is the muscular dystrophy. Muscular dystrophy is a syndrome that occurs due to genetic mutation and is characterized by progressive muscle weakness. And the Duchenne's muscular dystrophy. It is a mutation of the dystrophic genes, resulting dystrophin to be absent or from the muscles. It is a X-linked disorder found one in per thousand male infants. It causes a great disability and eventually proves fatal by the age of the 30 years. Now, another is Baker's muscular dystrophy. It is an autosomal recessive disorder. Mutation in the gene or for the sarcoglycan or other components of dystrophin glycoprotein complex. It is a milder form of the disease in which the dystrophin is present but is either altered or reduced in amount. Another is the malignant hyperthermia. It is also related to dysfunction of muscles ion channels due to the mutation of the gene of ranitidine receptors for calcium release channels in the sarcoplasmic reticulum. This mutation results in dysfunction of calcium channels to shut down after stimulation, thus more ability availability of the calcium causes increase in contractility of muscles, leading to the more production of heat. This is the malignant hyperthermia. These are the events triggered by the malignant hyperthermia. And this is the classical symptoms are hyperthermia, tachycardia, trachypnea, muscle rigidity, etc. These are the malignant hyperthermia. And another is the myotonia. Myotonia is a disorder that occurs due to abnormalities of the sodium and the chloride channels caused by abnormal genes on chromosome 7, 17, and 9 or 19. It is characterized by an abnormally prolonged muscle relaxation after voluntary contraction. And this is a myasthenia gravis. It is a disorder of the neuromuscular junction characterized by grave weakness of the muscle. And the normal muscle tone of a muscle may be increased or decreased, constituting an abnormal muscle tone. The metabolic myopathies occur due to the mutation of the genes that code enzymes involved in carbohydrate and protein metabolism of the muscles for the production of ATP, which is called the Macardal syndrome. Metabolic myopathies usually present as intolerance to exercise. So this figure shows how the muscle contraction differs in an unaffected muscles compared to one with the Macardal disease. Now, fibrillation and the denervation hypersensitivity. The denervation of skeletal muscles in lower motor neuron lesions causes placid paralysis of the muscles, fibrillation and the denervation hypersensitivity. Fibrillation is characterized by fine, irregular contraction of individual muscle fibers. And another is denervation hypersensitivity, which is refers to when the muscle becomes highly sensitive to acetylcholine. And another is the sarcopenia, which is age-related muscle loss. Now, I like to mention that these are the key regulators of excitation contraction coupling. First one is the neurotransmitters and hormones. Acetylcholine, epinephrine, and insulin-like growth factors, actually the main regulators, that is the neurotransmitter type or the hormone types of the regulators of excitation contraction coupling. Another is the ion channels and transporter. Voltage gated channels such as sodium, potassium, and the calcium. And another is the calcium ATPase. And third one is the signaling pathways, calcium calmodulin kinase and the protein kinase A. These are the key regulators of the excitation contraction regulators. Now, what are the potential therapies? The therapies are small molecule inhibitors. One is the ryanodine receptor blockers, and another is the calcium channel blockers. Gene therapy, dystrophin replacement, and another is the genomic editing by CRISPR-Cas system. And third one is the stem cell therapy. There is the muscle-derived stem cells and cardiac progenitor cells. Now, I like to summarize my lecture that excitation contraction coupling is the physiological process linking muscle excitation to contraction, initiated by motor neuron, leading to depolarization of the muscle cell membrane. Depolarization stimulates the sarcoplasmic reticulum to release calcium ions. Calcium binds to troponin, causing conformational changes that expose binding sites on actin by moving tropomyosin. Myosin heads attached to actin, pulls and release, resulting in muscular contraction. Calcium returns to the sarcoplasmic reticulum and muscle fibers relax. So this is the summarized view of the excitation contraction coupling I have already described in my previous slides. Now, excitation contraction coupling is a vital process that underpins muscle function, translating electrical signals into mechanical movement. A comprehensive understanding of its cellular mechanisms not only enhances our knowledge of muscle physiology but also illuminates potential pathways for therapeutic intervention. As research continues to unveil the complexities of excitation contraction coupling, opportunities arise for targeted therapies aimed at addressing various neuromuscular disorders and cardiac conditions. Innovations such as calcium channel modulators, receptor enhancers, and gene therapies hold promise for restoring normal muscle function and improving the quality of life for affected individuals. Ultimately, bridging the gap between the basic science and clinical application will be essential for developing effective treatments that target the intricate mechanisms of excitation contraction coupling. This endeavor highlights the importance of multidisciplinary approaches in tackling the challenges posed by muscle related diseases, paving the way for advancements in both understanding and treatment. So, these are my references, and thank you.
Thank you, sir. Thank you so much for your highly scientific and informative lecture. So we have some questions from the YouTube live. So I have one question: that is there any deficiency in the calcium in our body, that is human body, will affect in the EC coupling, sir? Could you please
Thank
highlight
you,
on
sir.
this question?
Actually, in my lecture, I have mentioned that the calcium is a bivalent cation and it is an important cation responsible for the excitation contraction coupling. If the deficiency of the calcium occurs, so it may obviously uh, 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 causes that the uh, in- excitation contraction coupling will not take place properly. So it is the one of the important issue. So proper calcium ion concentration is essential for the formation of the excitation contraction coupling. So if any subject is deficient from calcium ion, it will disturb the excitation contraction coupling, sir.
Okay. Thank you, sir. And another question from another participant. How does the ryanodine receptor and the DHPR receptor affect in Duchenne muscular dystrophy? So I think you already discussed about the receptor mechanism with the
Yeah.
myopathies.
I have
So
already
could you?
discussed in my presentation that the in case of the Dachinis muscular dystrophy that takes place, the ryanodine receptor and the dihydropyridine receptor that not function properly. When the action potential reaches to the transverse T tubules, a event occurs takes place in such a way that uh, the interlocking systems offend and the calcium is released. But in case of the Dachinis, the person suffering from the Dachinis disease or disorders, these systems actually is a failure system. So that is why the person suffering from the Dachinis actually uh, shows the various types of the physiological disorders. So it affects the system. So ranadine receptor and the dihydropyridine receptors interlocking systems may affect. It may causes due to the mutation or any other factors.
Thank you, sir.
Ultimately
And
affect muscular contractions.
yes, thank you, sir. One last question from another participant: that does EC coupling only occur in the skeletal muscle or any other muscle too? So I think you already
Actually,
discussed. Yeah.
I have stressed on the skeletal muscles, but this type of coupling systems actually also applicable for both the cardiac muscle as well as the smooth muscles, not
Thank
only
you,
for the
sir. Yes.
skeletal muscle.
Thank you so much again, sir, for your valuable time to share the highly scientific and informative lectures. So now I am handed over the session to Dr. Sandeep Mukherjee for the vote of thanks. Dr. Mukherjee, please.
Good evening, all, and thank you, Dr. Ornov, for excellent hosting. Today marks the 15th episode of online lecture series organized by the Education Committee of Physiological Society of India, and it has been both very informative and enlightening for physiology students. On behalf of the PSI Education Committee, I would like to extend my heartfelt thanks to our distinguished speaker, Dr. Bitin Kumar Maji, Associate Professor of Physiology at Sirampur College, affiliated to University of Calcutta, for delivering this month's online lecture. Thank you, sir. Along with his enlightening discussion on the AC coupling, Dr. Maji also explored its crucial clinical aspects, offering today's attendees a deeper understanding of its significance in physiology and beyond. His ability to connect fundamental physiological mechanisms with real world clinical applications made the session even more valuable. We are truly grateful for his efforts in broadening our perspective on this particular topic. I would also like to express my sincere gratitude to Dr. Prasun Priyo Naik, Dr. Rajan Haldar, Dr. Kausik Bharti, and Dr. Shampa Sarkar Vishwas for their invaluable support, guidance, and mentorship in organizing this particular lecture. And also a special thanks goes to Dr. Shampa Sarkar Vishwas for her essential technical assistance and ensuring the smooth execution of this session. Also, I deeply appreciate the support and cooperation of all PSI Education Committee members. And finally, a big thank you to today's moderator, Dr. Arnav Banerjee, for his excellent moderation of this session. And there is an announcement. Topic of the next month webinar is cardiovascular and respiratory changes during exercise. The esteemed speaker is Dr. Praveen Kumar, Associate Professor of Physiology at Tomo Riba Institute of Health and Medical Science, Naharlagun, Arunachal Pradesh. And this is from my part. So thank you all once again. Thank you.